see what we do here in the systems immunology laboratory, and this is the laboratory, and we do of course engage with a cohort of collaborators to get our research forward, and funding agencies continues to be DBT. Often time the funds do not come. And what do we study? We have two interesting focus: host virus interaction and also inflammation. Host virus interaction, I assume Jashi has alluded quite a bit today in the morning. And for inflammation, something that I will discuss here today. And what specific question, what is it that we are doing when it comes to inflammation? Let's have a look, because this is a multi-flavored forum I can see. So I'll try to summarize in a simplistic manner. What is it happening in during body when body is getting infected? There are these tissue resident cells. They are sensing these bacterial substances, and that would lead to activation of intracellular pathways, signaling pathways, that eventually will lead to activation of factors that would lead to gene expression. So this is intracellular signaling pathway. What I'm actually quoting here is this dynamical control, and this pathway would lead to eventually it will mount response to bacteria so that these pathogens can be cleared up. And the problem is it has to be intricately controlled. If you have too much of excessive of inflammation, you have too much of those signaling, it will cause tissue destruction. This is exactly what happened in COVID-19. Not many people died in COVID virus, but they died of the exacerbated inflammation that the lung has produced in response to those virus. On the other hand, if you do not produce adequate amount of inflammatory response, it will of course lead to immune deficiency. So this is the question: how is it achieved, that such an intricate control that inflammation portrays? Well, there are different groups. They have been thinking about it, of course. One part of this consideration is that cells in the body, they are in a tissue microenvironment. So when a tissue resident cell is exposed to microbial signals, they are also within a tissue where these other cells in the body, in that anatomic niche, continues to provide some additional cues. Some of them are tonic cues, they are body derived cues. They may not have to deal with directly with inflammation, and that modifies inflammation rate. And this has been a school of thought that microenvironment might be modifying inflammation, and we have taken that cue. Now again I am showing you this intracellular dynamic signaling pathway. S1 is nothing that instructive pathway that is activated in response to pathogen and leading to dynamical activity of signaling and transcription factor, and a particular expression of cohort of genes that will fight pathogens. And then these are those pathways that are ancillary, less important, we think peripheral, but they are nonetheless activated in the body because of tissue microenvironmental cues. And what we have learned in last 2 decades is that these pathways are interlinked. We have biochemically mapped them, we have established there are linkage, and this is where what we have been testing, the thesis, that this interconnectedness allows crosstalk between cell signaling pathways. And this cell signaling crosstalk between cell signaling pathways could be an important immunoregulatory mechanism. It might be important to tune inflammation to an extent that satisfies requirement in a particular anatomic niche. And this is the, of course, challenges with studying such cell signaling crosstalks are many. We are talking about two activating signals, and there are a lot of problems with it. We are going to talk about concomitant signals, sequential, which one comes first, what could be the dose, what could be the duration of each of these signals. And when these two signals would crosstalk in biology, it would lead to emergent properties, non-intuitive system properties, often difficult to track. Particularly, you would not know if the crosstalk effect would happen early or would happen late. And if you think about so many dozens of cellular pathways that we think about in biology, they are interconnected. That causes a lot of difficulty to intuitively predict mechanisms. You would have no idea. You would be feeling comfortable when pathways are insulated from each other, but in a network it becomes very difficult, it becomes challenging, and this is what people have done. They began to rewire these cellular pathways, signaling pathways, in a computational model, inform the model using quantitative biochemical analysis that pertains to cellular analysis, and then you can simulate variety of biological conditions, make predictable hypotheses and test those hypotheses in experimental settings. Thereby begin to understand how cell responds better, develop mechanistic cues and then test those mechanistic cues in animal model to see if it has any relevance, physiological relevance or pathological relevance. And we have been developing this elegant approach for studying signaling crosstalk at the National Institute of Immunology and trying to address its role in inflammation. But what are the pathways? We're talking about cell signaling pathways, immunoregulatory pathways, and this is one, the NF-kappa B system. It has two components. The canonical NF-kappa B pathway is this a magenta one I was talking about, the instructive pathway that gets activated in response to microbial signals, the magenta pathway. What you have here is this NF-kappa B activation in response to TLR or IL-1. This is a cytokine. TLR is the one that gets activated in response to bacterial stimuli. And you have a dynamical controlled activation of the canonical signaling, but oftentimes you have of course aberrant activity as well. And then you have this other NF-kappa B pathway. These are these microenvironmental cues in the tissue that would activate this pathway. That's the green pathway. It also has typically lymphotoxin beta receptor. These are, of course, has some other functions in immune development, not direct functions in inflammation or immunology. And it would lead to slow but sustained activation of NF-kappa B. And we have over 2 decades, we and others have shown these pathways, again the same analogy, biochemically interlinked. And that led us to question if crosstalk between NF-kappa B pathway is something that is important for regulating inflammation. So to understand crosstalk, as I told you earlier, that it is very difficult to figure out crosstalk regime, when it would happen, when it would not. So we developed a mathematical model. There are these two NF-kappa B pathways. We have modeled them. We have used experimentally derived kinase activity that are activated during this magenta signaling, canonical NF-kappa B signaling, plugged it into the model, derived NF-kappa B response. Similarly, we fitted the non-canonical signaling. So the model was working. So we have individual signal 1, signal 2. We are satisfied. We are going to now study crosstalk. And one result is from a very old paper, but I thought that this is to introduce what we are interested in. That was very instructive for the next decade of the lab. We used a theoretical kinase activity library. What you see here is all possible kinase activities, their durations, their amplitudes and their onset time. We plugged it in the model through the canonical arm, through the non-canonical green arm or both, and determined NF-kappa B crosstalk in the downstream signaling level. And we then just simply mapped what are the crosstalk proficient activities. One very interesting information came out that this kinases has to have a longer duration. It has to be sustained to engage into crosstalk. Looking at the wiring diagram, they are connected, but not always they crosstalk. The upstream kinases has to have a bit long lasting duration for them to engage in the crosstalk at the level of NF-kappa B signaling, and this is the experimental data. We immediately followed it up. The canonical, the instructive arm, the magenta signaling, they are activated by IL-1, it's a cytokine that typically give a very transient kinase activity, and TLR ligands like the bacterial ligands, they give a bit long lasting kinase activity. So we argued that in a crosstalk regime, when both the pathways are on, maybe IL-1 would not participate in the crosstalk and TLR ligands would. And this is an experimental data. What we are doing, we are activating cells by this magenta, by this green or both, and measuring in a nuclear NF-kappa B activity in an experiment called EMSA to figure out. You see this IL-1, there is virtually no difference when IL-1 was treated and when these two are treated together. But with LPS, you see there is an activity and there is a progressively deteriorating late phase. Once you co-treat cells with both LPS and non-canonical signaling stimuli, you see a progressive, this late phase is enhanced, suggesting there is a crosstalk going on. However, the crosstalk is stimulus specific. And within a year, and we actually went further. With using model, I'm not going to detail those. Using model, we figured out what could be the genetic link between these two pathways that is mediating crosstalk, NF-kappa B2 gene. It turns out, and we used a mice that lacks, we used a mouse model where NF-kappa B2 gene is deleted. And it turns out those animals, if infected with this Citrobacter rodentium. Citrobacter rodentium is a gut pathogen. If you infect those animals with Citrobacter rodentium, they do not elicit adequate inflammatory response for the lack of crosstalk, and they succumb to death. So it shows that immune regulatory mechanism, going back to the same word, so-called green pathway, which is of course not in the central stage, is tuning inflammation, ramping it up when it is required, when there is a pathogen challenge, and an absence of such crosstalk can lead to devastating immune failure. And within a year, we are going to macrophage, that are another cell type very important for immune response, and we didn't find any crosstalk in macrophage. What we did a lot of quantitative study to arrive onto a macrophage parameter ensemble, which also showed, very nicely recapitulated the crosstalk defect that we experimentally see in macrophage cell type. By exploring macrophage parameter ensemble, we were able to determine what could be the biochemical mechanism that separate these pathways in macrophages, presumably to protect us from deleterious overt inflammation that macrophage may produce. And this is something ongoing that we are doing, we have not published. This is experimental data. We usually do ordinary differential equation based deterministic model, but by introducing parameter variations, we have generated a model that captures cell heterogeneous NF-kappa response. In experimentally, if you treat cells with a ligand in a plate, not all cells would respond similarly. Some will show more response, some will show less response, and you can measure log variance, this NF-kappa response variance, not amplitude, variance. We actually used a model where we could recapitulate this log variance. Now see what we have discovered using that model. Very interesting, that this magenta signaling, of course you have log variance. So we are showing that if you signal through canonical arm, there is a cell heterogeneous NF-kappa response. The green one, when you signal through signal 2, the non-canonical arm, there is a cell heterogeneous NF-kappa response. The black one, we see that when you signal through both the arm, the log variances is going down, suggesting the signaling crosstalk not only impact duration and amplitude of inflammatory response, but may have relevance for cell heterogeneity, something whose biological, we have not tested this experimentally, we will hopefully do it next year. We do not even understand the biological significance of such variance control. And now just two more data from, I have 12 slides, although it looks a lot. One student of mine, Meenakshi, she took it a bit further. The insights that you gathered, that this crosstalk would be important for inflammation control. She went to study human disease, human inflammatory bowel disease. As you see, it's inflammatory, too much inflammation. What she found that these are IBD patients, and in their colon there is a recurrent activation of this green signaling, the non-canonical signaling, so-called naive signaling. It is activated during inflammatory bowel disease. And this activity correlated with inflammation, activity of the magenta signaling. So we began to say that maybe in inflammatory disease, this green signaling is fueling this magenta signaling and causes the disease. And immediately we did this animal experiment. In this animal experiment, what we are doing, we are treating animal with DSS, a chemical which will cause erosion of the mucin layer and epithelial layer, and thereby provoke intestinal inflammation. And then you collect those colon cells and measure the magenta activity. What you see in wild type, once you treat cells with DSS in animal, their colon, there is quite a bit of magenta signaling. But once you have a knockout where the non-canonical pathway is taken out, this signaling is going down. And then you use a mice where this crosstalk has been impaired in intestinal epithelial cell and treat them with DSS. Wild type mice, they are very sensitive to DSS. So you see huge disease index and they die by day 7 or 8. Knockout mice was less sensitive and they recovered quite well. Now what we are learning here is that crosstalk would be an immunoregulatory mechanism. It can protect from bacterial infection, but in certain settings where there is overt inflammation, it can actually exacerbate the pathology in the tissue. And then the last one that we are doing, of course we have matured as a group in over a decade or so. We are of course talking about signaling crosstalk. Now we are talking about crosstalk beyond the NF-kappa system, going beyond this cell signaling system that was our favorite because of Alvin and Navin. They looked into dendritic cells, another very important immune cell that determines how we respond to inflammation or pathogen. We used the mice, we used a dendritic cell which lacked the non-canonical signaling. What we see, there is an increased accumulation of beta catenin, which drove RALDH2 mediated retinoic acid gene synthesis, metabolic pathway, leading to a retinoic acid gene signature. And you know what is known about retinoic acid? If dendritic cells produce retinoic acid, it promotes regulatory T cells and it promotes IgA secretion by plasma cells. These two are good enough to restrain inflammation in the gut. So dendritic cell has an ability to control inflammation if you allow them to make more dendritic cells and this. So we began to question, is it important? Yes, in animal, now we are generated an animal where this pathway is selectively impaired in dendritic cell. What we see, indeed in this animal you have Treg accumulation, we have measured IgA, we see IgA accumulation, and IgA controls gut inflammation by changing gut microbiota. We see indeed there is a microbiota change. Firmicutes are good microbes. We see their increase in the knockout, and bacteria are bad stuff. Their amount is decreased. So it protected the ablation of the pathway in dendritic cell. The green pathway led to a better inflammatory control by altering Treg population and gut microbiota, and indeed these mice were far more resistant to colitis than the wild type mice. So where we are going now is basically going back to the slide I originally showed. You have pathways S1, S2, S3. Of course, you are very interested about S1. That is the principal pathway, this instructive pathway that controls inflammation. And inflammation sometimes good, sometimes bad. You do not know how to go about it. The problem with the knowledge is that you can't therapeutically target a hub pathway, a central pathway. It will cause devastating side effect because that pathway is not only involved in one thing, they are probably involved in 20 other things. And the second, one pathway centric approach doesn't allow you to even fathom how such intricate control, how such intricate tuning of inflammation can be achieved, right? So what we are saying now that these pathways are interlinked and variety of biochemical mechanism that allows tissue microenvironmental cues to tune inflammation in certain anatomic niche. They can ramp up inflammation, that can be protective in certain anatomic or physiological setting. Because they ramped up inflammation, it can have deleterious health effect. And second part of it is that they promise that you cannot target these hub pathways, but how about targeting cross-regulatory mechanisms that connect these pathways and thereby get a cell type specific, disease specific interventions. So we argue that our work that we have been doing, where we have almost established signaling crosstalk as a central mechanism, a dominant immunoregulatory mechanism, also bears promise for expanding therapeutic opportunities for inflammatory ailments. So, and this is I think it's a bit sketch. We probably have to do more, but I mean setting up logistic where you have multiple scale, multiple data analysis capabilities, and I more or less will thank you. What you have seen that we have been trying to do is combine different discipline, including mathematics, animal model, biochemical studies, to learn more about how immune controls are achieved, and hoping to take us to the era of systems medicine. Thank you very much. Thank you for your patience.
Thank you. Thank you very much. So, I'm sure there will be some, there's a beautiful talk, and I'm sure there would be some questions.
A very naive question. Do you think that the canonical pathway is early immediate reaction to say bacterial exposure, and then it initiates the non-canonical pathway into sustaining it on a longer
Yes.
level?
Yeah, the question is, yeah, I think it's a good question. You are saying that the canonical signaling, even if you fragment it into time zone, if the effect of non-canonical signaling would be more dominant at the late phase. That is true. That is what is happening. We think that you do not need crosstalk to get this early peak, but when will you fall will be determined mostly by this crosstalk. You might probably sustain a bit more, which is probably important for intestinal epithelial cells which are hyporesponsive in nature. But this is what we also see, we agree with you. And I think this is where the catch is, because some signaling is too transient. If it falls too early, then there would be not adequate immune response or neutrophil recruitment.
Ask is that, do you think that in individuals who are immunodeficient or who are highly susceptible to infections, it is possible that the non-canonical or the ancillary pathways are not as active, or they are not working when they are supposed to?
Yeah, so this is again good questions, right? But remember this, at least the non-canonical NF-kappa B pathway, this is important for immune development. This is important for lymph node development. And that's a big problem, right? If you have a mutation which is so severe to completely impair the non-canonical signaling, you won't have lymph node. And then the question that we are trying to address, will it also be important for crosstalk and thereby doing inflammation control become less important or difficult to address, right? More difficult to address. But people have mapped certain mutations in the non-canonical pathway. These are human data. Those were showing very surprising immune deficiency, despite those individuals having a full spectrum of lymph nodes. So something that we have not studied, were there inflammation deficiency that can of course impair immune system. But that's a good question, and thereby what we are doing in the animal model, we are using tissue or cell type specific knockouts. So I don't have to worry about the lymph node. Those animals will have lymph node. But despite that, if you delete the pathway in intestinal epithelial cell or dendritic cell, you see that some phenotypes are coming up. They are not because of lymph node deficiency. They are because of what is happening in that cell type, what we call cell intrinsic signaling problem.
So non-canonical pathways in many cases usually diversify a cellular response, but in your results when you put both canonical and non-canonical, the heterogeneity was least. Does that mean it's there in this case to reinforce rather than diversify? Is that an interpretation?
This is good question, great question. Actually, I have to emphasize here, when you say diversity of the response, one aspect of it is that gene response, let's say. I am not ruling out that when these pathways operate together, gene response will be indeed diversified. We don't have any evidence suggesting that it would not. What I was trying to show you, it's a simulation results. It's nuclear NF-kappa B activity heterogeneity. In some cells you have 10 nanomolar, some cells you have 100, despite they are in the same plate. What we see that of course when you give the canonical or non-canonical, regardless of the amplitude of the response, the heterogeneity was similar, the central variance was similar. When you simulate that where both of them are together, you see the heterogeneity is going up. Now it, in conjunction with the fact that this non-canonical signaling amplifies canonical response, we begin to get somewhere. Maybe you are amplifying response and making it synergistic to have a rapid response to pathogen that innate immunity demands. But this is a complete conjecture, wide ideas.
So, thank you very much.